Thank you for coming here. I'm going to present over the next 15 minutes a little bit about the rocket story. First, just a safe harbor statement, as we're publicly traded, so there are going to be some forward-looking statements made here. But one of the first things I want to take, if there's 3 takeaways about Rocket, I'd say, one, what's unique about Rocket is every one of our programs is intended to be first, best, and in some cases only in class. So we carefully chose our pipeline candidates that would meet that strict criteria. Second, we're in a transition to commercial. We announced this past week a acceptance by the agency on our first BLA, and we have a PDUFA date of end of March, and we have a second filing that's underway right now. So commercial readiness activities are underway. And third, we're very well capitalized, and we have a team that has a proven track record of executing with regards to operations in cell and gene therapy. So underpinning a lot of our goals, and if you come to either of our offices, you'll see that we have these 4 core values. And they're actually, when you think about gene therapy and the word gene, it actually shares the same root to the word generosity. And when you have a mission where you're trying to pursue cures for devastating diseases where you're first, best, and possibly only in class, these values are very important, because if you don't pursue these cures for diseases like LAD or Danon, no one else is, no one else will. So it's incumbent upon us that we always reiterate the value system of trust, curiosity, generosity, and elevate. Elevate because some of us who joined when we were under 10 employees, we've built this company to close to 300, and we're here to stay. So with regards to a high-level overview before we start going to our programs, we're a multi-platform company. We've built a facility that's close to 103,000 square feet. On the heels of the alignment with the agency on our pivotal trial for Danon disease, we raised an additional $200 million last month, co-lead Morgan Stanley and JP Morgan, and that provides us a solid balance sheet to take us through the end of 2025. And we continue to add to our executive leadership team that has a track record of more than 20 drug approvals, many of which that happen to be in the biologic space, which is the closest analog to cell and gene therapy. Now, with regard to that first point I mentioned on having a pipeline of being first, best, and only in class, when the company was formed in July of 2015, gene therapy was largely focused on the autologous lentiviral space. And so those 3 programs that we initiated in 2015 and 2016 are the ones that, 6 to 7 years later, are now advancing to filing stage. So when you see the top 3 columns, the LAD1, which is the smallest of our indication, is the first one which has a line of sight at approval with the PDUFA date of March 31st. Fanconi anemia, and then pyruvate kinase deficiency, which are also blood disorders. And each of those 3 are late-stage candidates. On the heels of that, the pipeline has 3 additional AAV heart failure candidates. So our pipeline is not only diversified between heme on the rare blood disorders, but also 3 AAV heart failure indications. And when you look at the cadence of approvals and launches over the next 5 years, each one of these has larger and larger addressable markets. And so you can see a lot of the regulatory designations, and now with Danon disease, with an agreement to initiate the pivotal trial and the PKP2 already in phase 1, we're hoping that those programs and the BAG3, which we acquired from RenovoCor last year in that acquisition, those 3 should follow suit as well. As I mentioned, these two franchises that we have, we're one of the few gene therapy companies that's platform agnostic. We consider ourselves from the founding to be a rare disease company. So we chose assets where we could make sure that, thinking about the central dogma, DNA, RNA, protein, that we could target the cell of interest, deliver the gene of interest so that we can produce the protein of interest. And if it's a blood disorder, which happens to have rapidly dividing cells, the ex vivo platform with the lenti backbone is suitable. If it's a solid tissue, such as the heart, where, as you know, cardiomyocytes don't have any turnover after the several weeks of birth, the AAV platform is suitable. So we make sure that we have a strong science background that marries the properties of that platform with regards to tropism to choose which platform, and so we're agnostic. And in the future, if there happens to be another platform and we find a disease that is worthwhile targeting, we will pursue that. On the lower left-hand corner, you'll see a small picture of our Cranbury, New Jersey facility. And this was an endeavor starting from the middle of 2018 when we recognized that for some of these larger markets, we will need dual sourcing. So we made a concerted effort to bring some of these functions in-house. And in that facility, the primary focus at this time is AAV viral vector production, as well as in-house analytics, PD, and currently out of about 280 people, I'd say 250 reside primarily in Cranbury, New Jersey. We still keep our headquarters in New York City in the Empire State Building. And with regards to these two platforms, we also have alignment with leading CDMOs in US and Europe, especially on the lentiviral and cell processing platforms. But at the end of the day, the goal, as you see on the right-hand side, is to maximize the patient reach. We're cognizant of the high cost of capital. In a perfect world, in the large pharma sector, you would have dual sourcing across the supply chain. But as our programs get to commercial, those are the kinds of things that we will evaluate with regards to both drug product and drug substance. But with the in-house capability, we can accelerate a lot of the development, especially for our Wave 2 pipeline, where the internal goal is to go from IP filing and initial target identification to an IND in somewhere approximating 24 months. And with an in-house R&D team and PD capabilities, we can easily migrate from that to GMP manufacturing, where we've already been producing registration batches in that Cranbury facility. The first indication I'll cover is Danon disease. So last month, we arrived at alignment with the agency, but maybe a little bit of an overview. This is a serious unmet need indication. Dr. Danon actually came across this indication in 1981, but it was several decades before the literature really started to pick up the severity of this disease. This is an X-linked dominant disease that's monogenic in nature. It's a disease of autophagy. So when you think about the heart, which has the LAMP2B functions as like a, I think the analogy is you have these, like a vacuum cleaner. And unfortunately, if you're not getting enough LAMP2B expression, you get these garbage bags that start to accumulate in the heart. And the heart, over time, gets larger and larger and larger until at some point, a heart transplant's needed. So if you look at the literature in the Kaplan-Meier curve, the median survival for these boys is about close to 18 or 20 years of age. And a heart transplant, even with that, has its side effects. So therefore, this was a disease where gene therapy, especially in a loss-of-function mutation, was ideally suited if you can deliver the LAMP2B gene and it can stay in cardiomyocytes. As I mentioned, because those cells don't have turnover, unlike liver cells, if you can effectively deliver the DNA into those cardiomyocytes, and they stay there for ideally decades, then you can start to produce the LAMP2B, and it remains to be seen exactly how these children will do. But as I'll show you in the efficacy slides, our thesis has held true to date. So this, you can call it a heat map. These are all the patients that we treated in our phase 1 trial. So across the columns are, on the right-hand side, a number of different endpoints. Many of these endpoints, these biomarkers, are highly indicative of clinically relevant endpoints. And as you can see in the green, there has been improvement in the dark green and in the light green stabilization. There are no boxes that are red, so there hasn't been any deterioration or worsening in these patients. So we know that this, at least from these n=6 patients, that there has been improvement and an effect, a positive effect, from delivering the LAMP2B gene. Of note, you'll see that some of these patients, the early patients, have now been followed by close to 3 years, at 36 months. And there's been improvement also on the volume, as we've highlighted in bold, the LV mass. The reason I'm mentioning this is, as I go to the following slide, we recently pursued the agency on an alignment for a registration trial. So we arrived at an alignment last month for a single-arm study that is robust and very efficient, with 12 patients, with 2 co-primary endpoints, the first being protein expression of LAMP2B, and the second, a very clinically relevant endpoint of LV mass, left ventricle mass of the heart. And I'll explain why in the following slide, but those 2 endpoints, if you follow out at 12 months, should give us a positive readout and a chance at accelerated approval. And what we had already previously discussed leading up to that was the other elements of the submission, including CMC and analytics. So our Cranberry facility, we've already made our registration batches that are going to be used in this trial, and several sites are already up, and enrollment is underway. Now, with regards to that second endpoint, LV mass index, taking a step back, Danon disease, and you can't lump all heart failure in the same way, but Danon disease simplistically is a disease of big hearts. If you look at the literature, some of the biggest hearts on record have been patients that have been afflicted by Danon disease. So if you can get echoes and you can actually track the volume over time post-treatment and you see a reduction, it's a clinically meaningful endpoint. And if that's also supported by New York Health Classification and other endpoints, and these patients are living into their 20s, it's highly indicative of something that's efficacious. And as you can see in the lines on the left, many of these patients have been followed out, several of them close to 3 years. And we reached agreement with the agency of at least a 10% increase, 10% decrease of left ventricle mass index. Go to our next program, which is Fanconi anemia. And with regards to Fanconi anemia, this is a disease of DNA repair, and these Fanconi anemia afflicts between 5,500 to 7,000 patients between the US and EU. Fanc-A is the largest complementation group, close to 65%. And this is a disease where, despite the ability to get bone marrow transplant, allogeneic transplant still has a risk of head and neck cancer of 50% at 10 years. So therefore, something that can produce efficacy here and avoid that transplant is definitely worthwhile. We've also shown the endpoint here with regards to hitting the endpoint, 5 out of 12 patients exceeding the MMC resistance. I think I'm slowly running out of time, so I'm going to try to flip through the other slides. LAD-1, this is the agreement that we reached with the agency for a BLA acceptance. And this median survival here, according to natural history for these kids, is about age 3. It's severe LAD, has CD18 expression of 2% or below. And in treating the 9 kids, we had 100% survival in 9 out of 9 kids, and our protein expression is well north of that. So the agency gave us a PDUFA date of March 31st on this. Pyruvate kinase deficiency could be the largest of our lenti indications. Similar to beta thalassemia, many of these kids are of transfusion dependence and are post-splenectomy. We treated some of the worst patients that are out there that are highly dependent on transfusions, and we doubled the hemoglobin from 7 to 14 grams per deciliter, and these patients are now living a normal life. While there is an approved product for early stage by Agios, our goal has been to, at the current time, to pursue this for severe pyruvate kinase deficiency, but it could be the largest of our lenti indications. And then we recently announced an IND clearance of what we call our Pegasus program, PKP2, which could be the largest of our programs, more than double the size of Danon. And this is actively enrolling in phase 1 at the moment. And we think this can be first and best in class, not just with the obviously first, but we think that while this is a gene therapy product that has multiple market entrants, learning of the success that Sarepta and others have shown in Rh74 versus 9, we think that especially if you have to go to adult heart failure, we have a median age of 35. We think that Rh74 could provide a wider safety window. And we optimally designed a vector here with the cardiac-specific promoter, a similar route of administration, and a lot of our know-how, and we have very strong proof of concept here. Finally, as I mentioned, that we acquired Renovacor last year, so BAG3 is another large opportunity in heart failure, and we're currently in IND-enabling studies, and this is anticipated to be our 3rd clinical program in the near term. So I guess the takeaway, as I mentioned, Rocket has a preeminent, now maturing pipeline where everything is first, best, and in some cases only in class. We have a diversified pipeline that's equally distributed between lenti and AAV. We are transitioning to commercial with an experienced management team that actually has a history of delivering. And we're well capitalized based on the recent financing, so we can get at least through the next 2 years.